Good evening. It is my pleasure to welcome all the participants to this international webinar series on the Trinity of COVID-19, immunity, implication, and interventions organized with the Department of Biochemistry and Microbiology. Severe acute respiratory syndrome coronavirus 2, called as SARS-CoV-2, is a causative agent for the ongoing coronavirus COVID-19 pandemic. There is a sharp rise in the number of new cases emerging from India, and we are yet to reach the peak. Efforts to overcome the virus is hampered by a lack of knowledge on several important aspects of SARS-CoV-2 infection, ranging from pathogen biology to the host response and treatment options. Therefore, there is an urgent need to better understand the host pathogen biology of COVID-19. I'm sure this international webinar by prominent scientists and researchers will give an insight to the ongoing crisis. I'm very happy to welcome the speakers, Dr. Gokul Shankar, Senior Associate Professor, Faculty of Medicine, AIMS University, Malaysia, Dr. Gauri Shankar Rajam, Associate Principal Scientist, Merck & Co. USA, Dr. Padmavati, Professor Sri Balaji Hospitals, Chennai, Dr. Naveen Kumar, Founder and Managing Director, Immunogenics Biosciences Private Limited, Chennai, who will all be sharing their expertise in the 3 days. I thank all the speakers for accepting our invite. I would like to thank our correspondent, Dr. Lata Rajendran, and Chairman Mr. Kumar Rajendran for their encouragement and constant support. I request Dr. Durga, Assistant Professor, Department of Biochemistry, to introduce our speaker of the day, Dr. Gokul Shankar. Over to Dr. Durga.
A very good evening. It's a great pleasure for me to introduce our speaker for the day, Dr. S. Gokul Shankar, Senior Associate Professor, Microbiology Unit, Deputy Dean, Students and Alumni, Faculty of Medicine, AIMS University, Malaysia. Dr. Gokul Shankar has his doctorate in the field of microbiology, armed with an extensive career of more than 14 years in research and a distinctive expertise of more than 7 years as faculty in microbiology for the postgraduate and undergraduate students in India and Malaysia. His expertise includes planning, coordinating, and conducting research work on microbiology, claim authentication for cosmetic and personal care products, development of herbal and cosmetic formulations and food supplements, demonstration of extraordinary microbiological techniques, and conducting and coordinating clinical trials. He is also extremely conversant with various aspects of food, pharmaceutical, cosmetic microbiology, and mycology. Dr. Gokul Shankar has published several research papers in top national and international journals. He also serves as a scientific reviewer for many reputed journals such as Journal of Yeast and Fungal Research, Journal of Medicinal Plants Research, Journal of Basic and Clinical Pharmacy, Journal of Bacteriology, and so on. He is also an approved research supervisor for guiding PhD programs at Acharya Nagarjuna University, Andhra Pradesh, Satyabama University, Chennai, Karpagam University, Coimbatore, and AIMS University, Malaysia. Dr. Gokul Shankar has received funding under the Fundamental Research Grant Scheme, Malaysia, for his project entitled Gene Knockout Studies of Non-Protein Coding RNAs as Potential Candidates in Regulation of Biofilm Formation in Salmonella enterica. He also has several honors and awards to his credit. To name a few, Young Scientist Award awarded by the University of Colombo, Sri Lanka, SAB Award of Excellence in Food Safety, 2016 Award of Excellence in Food Microbiology, and Q1 Star for Excellence in Overall Academic and Research Performance awarded by AIMS University, Malaysia. He also has been awarded the Best Teacher Award 2019 from the Microbiologist Society of India. We are extremely grateful to have an eminent personality amidst us today, and I request Dr. Gokul Shankar to share his knowledge and expertise with us. Before I hand over the session to Dr. Gokul Shankar, there are a few instructions for our participants. Participants, kindly post your name and college name in the chat box. And towards the end of the session, kindly post your queries to the speaker in the chat box. Thank you. Over to you, sir.
Good evening, everybody. It's nice to be with MGR Janaki Amman College once again. Thanks to the organizers and the management of MGR Janaki College. Okay, today my topic is a very simple one but interesting one. It's not on COVID, but it's something on how to build your immunity. So they say germ is life. Today, our lives are totally changed by a tiny, tiny virus. It has made all the normal behavior to be out of normal. Okay, I remember a popular joke which says like, before the COVID scenario, if anybody sneezes, we used to say, bless you. Now it will be get lost or say no manners, you're sneezing in public. So germs have affected our life in every way. Today's talk is actually about our gut microbiome, which affect our lives, and what are the simple things which we can do to keep our gut microbiome happy. Can I have the slides, please?
Sir, please open your PowerPoint, sir.
It's open only. Just a minute, I'll just sorry. Should I stop share and start it again? I'm not able to see the. Everybody, hi. Can you see my slides? Okay, is my slides visible?
Yes, sir, it is visible. Sir, it is visible, sir. Please
Okay.
proceed, sir.
Thank you. Sorry for the break there. Okay, so we need microbes to be programmed for good health, and we need the right combination of microbes in our lives. So over the years, if you see, like we have adapted to coexist with them, and we have started acquiring them at birth. So we should also acquire them at a right combination. So we always have a feel and pride that man is in the top of the evolutionary ladder. So, but if you see the genes which are there in the human being, like it ranges between 20,000 to 25,000 genes, which is much smaller than uh, a grape wine, or it is much, much smaller than a water fly, the genes. So we need to feel humble that for example, the rice plant has got more genes than us. But what keeps us in the top of the evolutionary ladder is the microbiome, the gut microbiome, what we have. So we have just over 25,000 genes, but the gut microbiome contributes the remaining. We have 100,000 genes, which is that of the gut microbiome. So that makes us really a super being. Now the question comes in, how human are we? Human cells, the number of human cells, what we have in our body is 10 trillion. And what we have as microorganisms, the number of microorganisms in our body is 100 trillion cells. And if you compare the genes, what we have and what they have, it's 23,000 genes. Okay, and the microorganisms which are there in our body, they have 3 million genes with them. So actually, every human being is a supra-organism. And it's a environment and a lot of other factors which govern the interaction between us and the microbes. And there is a delicate balance which exists between the microbes and us. If this balance is altered for any way, okay, it's going to affect the health status of us. So there is a little transition on the diversity of these microorganisms will transfer from normal good health to a disease state. So as I told you, we are 90% microbial, and we have over 1,000 different species of bacteria, not just bacteria, even other microorganisms in us. So we started our learning on our microorganisms, especially the gut microbiome, after the start of the Human Microbiome project. So you know that different body sites have different types of microorganisms, and there are different organisms which are predominant at different sites. But if you do a fingerprinting, it's not just a different site. Okay, for each of the site, for each of the individuals, there is a unique fingerprint. Means like each of your microbiome is unique for you. So that's something which is like your human chromosomes and your genetic, your like your fingerprints and other things. Your microbiome is also very, very unique for you. So as I told you, it's not just the bacteria. You have a lot of fungi, which is also there in your body. Okay, and some of them are also in the gastrointestinal tract. You have a lot of viruses as well. Okay, and some of the predominant fungi are the Candida and the Aspergillus species. Okay, now when we start acquiring these microorganisms, actually it starts at birth. Right from the time of birth, we start acquiring the microorganisms. So when it's at birth, is there a difference between a normal vaginal delivery and a C-section? Yes, there are a lot of difference which is going to affect the diversity of the microorganisms. So when it's a normal vaginal delivery, you can see like there are a lot of bacteria which is getting added to the gut, which are fecal in origin, which are vaginal in origin, and you also have some skin bacteria coming up. But if it is a baby delivered by C-section, it is just the skin bacteria of the mother which is getting added. So there is something in this beginning of the life itself, a C-section baby is lacking, is that there is a lack of diversity of these microorganisms. So then when you ask like, is it we need to avoid C-section altogether, but you need to also consider the fact that C-section is it's not an option always. like sometimes it becomes inevitable because it's a life-saving measure for a mother and the baby. And there are, besides this, there are a lot of other things which also govern the uh, microbiome of the uh, infant, such as breastfeeding, antibiotic treatment which the mother or the baby has received, the number of pets, like, or the presence of pets in your home, the number of siblings there are there in the home, the level of hygiene in the home, and also the nutrition of the mother and the child. Okay, when I say hygiene in the home, it should be remembered that it is sometimes better. It is not that perfectly clean all the time. So if it's not that particular perfectly clean, you keep adding more microorganisms which help to get the diversity. So there are a lot of research which are going on currently to know that the differences in the diversity or the predominance of a particular group of microorganisms may have an implication on certain diseases. Say, for example, certain allergies are predominant when you have proteobacteria with this normal microbiome. So the dominance of certain bacteria at certain conditions can result in an altered disease state. Okay, so then you have a question like if it's a baby which is delivered through C-section, then what's the way? Okay, as I told you, it's not just a C-section. After the C-section, there are a lot of other environmental factors which contribute, which also includes the breast milk. So something which is very fascinating about the breast milk is you have some components which are called as human milk oligosaccharides. It's called as HMOs, which are present in the breast milk. But it was a puzzling and a mysterious uh, role of these substances because they are not digestible by the infants. So for a long time, people were thinking like why these substances are present, okay, in the breast milk, when why the mother has to produce these human milk oligosaccharides when it is not digestible for the baby. But later, it was a great revelation that these substances are actually produced to feed the friendly microbes in the gut. So now the question is, how well do we know about our gut microbiome? So if I ask you a question to name a bacteria in your gut, most will right away say E. coli. Is it not? Because you have heard about E. coli so many times, and you are so um, knowledgeable or about E. coli that you just think of E. coli first. But what is the truth is, like, there are a lot of other organisms which outnumber E. coli in a number of 1,000:1. Means like there are 1,000 organisms of the other organisms when compared to E. coli, which is just one. So there are organisms which are like bacteroids, Prevotella, and so on, which actually have a major role in the gut. So it's not as simple as it's. If I have to explain it like this, if you have to plant a blade of grass for every bacteria which is present in your gut, then you have to plant grasses in the fields, in the football, in millions of football fields. So that much is the abundance of the microorganisms in your body. Okay, so we have actually co-evolved with these microorganisms. So if you see, from the time man evolved, he has been harboring these microorganisms. It's an evolutionary importance because the host, which is the man, and the microbiome, they had some common interests, okay, and neither of them they wished harm to each other. So they wanted to be with each other, That's the reason, if you see, in most of the bacterial symbionts, which is that in your gut, they lack certain genes, which are very, very important for them to survive in the environment. They have lost the critical genes, these bacteria which are there in the gut, to survive separately outside the gut. At the same time, they have retained certain genes which are of no use to them, but they are more beneficial to the host. So all the biosynthetic pathways the gut microbiome takes is actually a sort of an extension of the host metabolic and physiological activities. So we were using these terms microbiota and microbiome. So to get a better understanding, all the organisms the which are present in your body, okay, the collective organisms, you can call them as microbiota. And then the collective gene of all these microorganisms in your body, you can call them as microbiome. Okay, and also you will also hear a term dysbiosis, which is quite commonly used when we talk about gut microbiome. You have heard about the term symbiosis, right? Symbiosis means like the mutual relationship. Same way in dysbiosis, there is a dysregulated communication between the gut microbiome and the host. So there are a lot of diseases which are implicated with these dysbiosis, the common ones being diabetes, colon cancer, okay, and irritable bowel syndrome. So you know that each of these microorganisms are specific. So even if you talk about the gastrointestinal tract, there are different parts, like like stomach, duodenum, you have the colon, you have the ileum, jejunum, and all that. So you have different set of microorganisms which colonize each of these categories. So if you see here, you have the Helicobacter pylori. Helicobacter pylori is also a commensal flora. Okay, but why it causes ulcer is because of the dysbiosis. So it's a commensal flora. So, but when the numbers increase or they are dysregulated, then they can cause a infection. Now, there is a interesting component in this data which was presented from the American and British gut project. Okay, so they studied the gut microbiome of the people in the United States, in the United Kingdom, and other countries. So what they found was the diversity of the microorganisms was very less in the United States, relatively okay in the United Kingdom, but when compared to the other countries, especially the Asian countries and the African countries, you see there is a lot of diversity of these microorganisms. So what they found in this study is like the diversity of the microorganisms which are present in the gut. It also depends on the different plants eaten per week. When I say plants, it also includes the vegetables. So that is a key factor. So when I say it is a variety of plants, it is not that it doesn't matter whether you are a vegan or a vegetarian. It means like even if you are a vegetarian, you are eating a similar type of a plant product. It means like you are eating potato 3 times in a week or 5 times in a week, then that is not going to contribute to your diversity in the microbial flora. So what they say is like at least you should have 30 different plant items in a week. Okay, so that is to help you to diversify your gut microflora. And usually, if you see uh, people who are diabetic, they will be using artificial sweeteners, and in the sense they don't want to increase their sugar level, so they use artificial sweeteners. But if you see, they have tested artificial sweeteners on the mice models. Okay, they have used artificial sweeteners on thin mice. Okay, and they found that they have a tendency to become fat and diabetic. So they just wanted to know like whether it is just the sweeteners which are contributing to this or anything else. So what they did is like they isolated the bacteria, the gut bacteria from these fat diabetic mouse, okay, which is a fecal transplant, and then they introduced into the normal thin mouse. So they saw that the normal thin mouse, just with the introduction of the fecal transplant of a fat diabetic mouse, is able to change a thin mouse into a fat diabetic mouse. So what we were thinking all these days is like using an artificial sweetener is going to help you in diabetes. In fact, it may worsen the situation. Okay, the artificial sweetener may actually give you diabetes. So this is an animal study that we have not done much studies on human beings, and this is another interesting project where there is an association of the bacteria with obesity. This is a project in which a lot of identical twins were taken into the study, and they were followed up for a period of almost from the time of birth to almost 30, 40 years of age. So in this particular twin, what they found is like out of the two people, like one of them was thin, other one was little obese, and they found that the gut microbiome is different because otherwise, because they are identical twins, both of them they had the same genetic makeup. The only difference was in their gut microbiome. They found that certain bacteria, uh, which are Cellula and Akkermansia, which are more in the lean person. So if you see this, they have done experiments on this, like from an obese person, they have taken a fecal transplant, okay, and they have injected it in germ-free mice. So they saw that the mice becomes obese. But at the same time, if this fecal transplant is given with a co-culture of Cennella, okay, then the mice develops into a thin normal mouse. So the point is like it is a gut microbiome which affects the obesity or a person being lean. So this answers most of your queries. Like you may see your colleagues, some of them thin. You will see them that they are eating more, but they are still thin, and the other person who may eat very less may be obese. But that is not to do with the food, but it is because of the gut microbiome. I remember during my college days, one of my friends who used to eat 28 to 30 idlis in a day. Okay, he used to be very lean. So I was talking about Akkermansia. So if you see Akkermansia uh, in the obese or diabetic person and the thin person, then the difference is like you can see that the thickness of the mucus layer is reduced, and then you can also see that the inflammation is more. Okay, and then when you have these Akkermansia, they actually sort of protect. How they do it is like they alter the gut barrier function by increasing the plasma IBS levels. Okay, and that by there is a low grade inflammation. So something which is interesting in the other study, which I'm going to talk about now, is like this is again a study which involves the twins, where there were two people, twins, identical twins, and they were given the same food, but the response is different. The sense uh, both of them liked Prosecco. Prosecco is a type of white wine. So they were given Prosecco, but the blood glucose level spiked when Prosecco was given in one of the twins, but in the other one, there was a plateau. Whereas in the first one, when rice was given, it was a flat curve, and rice actually raise the blood glucose level in the other one. So different people actually respond to the food differently. Means like it can be the same food, but people will respond differently. That is because the gut microbiome of each of these individuals is unique. We have heard a lot about this red meat being bad and all that. Why is it bad? Is like you have this production of TMAO, which is the trimethylamine oxide. So what happens is like when you take the meat, you have a lot of carnitine and choline in the meat products, which is degraded by these gut microbes to produce this TMAO. Okay, what happens because of this is like this helps to build up the fat in the arterial walls, which can result in arteriosclerosis. So indirectly, it can affect your heart. So that's the reason they say like the red meat is bad for health. So there are things which are positive side to the microbes, and there are things which are negative side to the microbes, which are there in our gut. If you see like what they normally do is like they sit in the gut, they keep on doing fermentation. So the amount of alcohol which is produced in the colon every day, it's equivalent to 1 can of beer every day. Um, but good thing is we don't get tipsy out of it. And what else they do is like they produce a lot of short chain fatty acids. Okay, and we'll see what is the use of these short chain fatty acids later. So, but the negative effect is like when there is a dysbiosis, then you say that these organisms can cause different autoimmune diseases like ulcerative colitis, irritable bowel disease, and so on. So, if you see this picture, almost every disease uh, which is known, which is a non-infective disease, is known, maybe attributed to these gut microorganisms. So a lot of animal studies are being done, and if you see the uh, literature, there are a lot of new studies which are done every day, okay, to establish the link between the gut microorganisms and different diseases. So what we need to do to favor our gut bacteria is like we need to have the right amount of refined carbohydrates, and we need to eat a lot of vegetables, legumes, whole grains. Okay, so they are the prebiotics. Okay, we'll discuss on prebiotics a little later. And then the fermented products like yogurt. Okay, and healthy fats, which means like olive oil, nuts, avocados, and all that. Okay, and high quality protein. So this is a picture which shows the barrier between the microorganisms, the mucus, and the human gut. So what these organisms do there is like when you feed on a fiber-rich diet, that's what we were talking about, diet which is rich on plant products. So what happens is like these organisms uh, utilize all the fiber-rich diet and produce short-chain fatty acids. But if there is no fiber-rich diet, what they do is like instead of feeding the diet, they start feeding your mucous membrane. So you can see that there is a depletion of the mucous membranes here. So which can pave way for infection by different pathogenic bacteria. So if you see here, the fiber is digested by the gut bacteria. They produce a lot of these short chain fatty acids, which are the acetates, butyrates, and propionates. And what is the role of these short chain fatty acids is like they are the energy source for the luminal colon cells. Besides being the energy source, what else they do? They also have a protective effect. It's more important in the immunity, okay, because they protect against a lot of enteric pathogens. How they do it is like, if you see like these short chain fatty acids, they can regulate the virulence of certain pathogenic bacteria. Say, for example, Listeria monocytogenes. Okay, it produces uh, a toxin which is called listeriosin. Okay, it's a pore-forming toxin. The production of this toxin is reduced, okay, when there is a lot of short chain fatty acids, okay, in the gut lumen. Okay, similarly, like Salmonella enterica, uh, it has a virulence factor which is called a type 3 secretion system. I don't know whether you have studied about type 3 secretion systems. Like it's a sort of uh, injection or an injectisome which the bacteria has. It has a virulence factor. So the expression of the type 3 secretion system is reduced when there is butyrate in the gut. So it's time for us to reflect and think what probiotics and prebiotics would be helpful to produce a lot of this short chain fatty acids in our gut. So besides the other diseases which we discussed, where the gut microbiome have a definite role, there is also a link that the gut microbiota is linked to the mental health. They are found in studies like that. For example, in one of the studies, what they did is like they took the microbiota of a depressed patient and transplanted it to a germ-free mice, and they found that the mice also had depressed or the depression-like behaviors. So what is very interesting is like in some of the human studies, they also found that the stress can allow the bacterial antigens to cross the bowel barrier. Okay, and a lot of researchers believe that there can be the association of these gut microbiome in diseases like Alzheimer's and mental health. Okay, so when you talk about the, it's called the gut-brain axis. Okay, so there is a link between the gut and the brain. So in a healthy state, when you have a normal gut microbiota, so you have a normal digestive system, your mood is normal, your behavior is normal, even your immune responses are normal. But when you are in stress, that is because of the altered gut microbiota, because of the dysbiosis of your gut microorganisms. Then you have an altered digestive function, you have an alteration in the mood, okay, you have an alteration in the behavior, you have an altered immune response. So mood is directly related to food. Those days, people believe that different food had different vibrations. They call it as good vibes and bad vibes. But now we know that these vibes are nothing but the food which can influence these growth of the gut microorganisms. Okay, so in one of the other interesting studies, what they did is they tried to see the stress level and diet, whether they are correlated. So this was a study which was done on young volunteers, mostly school and college students. So they studied the stress reduction in these with the dietary change. So in one group, what they did is like they took a group of students who were stressed. They were just counseled and they were given advice. Okay, in the second group, they were just they were counseled and also given an advice to change their diet. It's like the diet change was like to include yogurt in the diet for at least 2 times in a day and eat a lot of vegetables. Okay, including the green and leafy vegetables. At least in a week, they were advised to have 20 different varieties of vegetables, and this was a study which was done for a period of 30 days. They found at the end of the study, they found that the group which received just the counseling, they still had the stress as such, but the one group which received the counseling along with the dietary change, okay, they became near normal. Uh, this is another interesting experiment done by uh, a scientist who's very famous for his gut microbiome research. He's called Tim Spector. Okay, he did an experiment on his own son. What he did is like he asked his son to volunteer to participate in a study with his friends. So the condition was they have to eat McD food, McDonald's food, okay, for a period of 20 days, okay, without any change. Means like morning McD, afternoon McD, evening McD. All 3 times eat McD, okay, for a period of 20-25 days. So what they found is like they analyzed the gut microbiome of all these participants before the start of the study. And at the end of the study, they found that almost 60% of their diversity in the gut microbiome was seen. Okay, so there was 60% reduction in the diversity was observed. So what we are trying to imply here is like, so when you take a proper food, you maintain a diversity of microorganisms. I want to compare this with the tropical rainforest. But if your food is not proper, then you are going to create or select only a few uh, dominant microorganisms to flourish, okay, and not a diverse group, then it's going to be looking like this, like a desert. There is always this question on whether there is a solution for Clostridium difficile infection. So this is also called as antibiotic-mediated diarrhea. So literally one of the things which modern medicine couldn't do much is like the Clostridium difficile infection, because it's not very successful with the treatment, because with the anti microbials which we use, because it is the antibiotics which are responsible for the antibiotic associated diarrhea. So what studies have done is like they have developed what is called as frozen poop capsules. It is the fecal transplant from healthy donors, healthy volunteers. So they have isolated the uh, gut microbiome, healthy gut microbiome from the healthy volunteers, and from the poop, means like from the fecal material, and they have made capsules out of it. And this, by taking these capsules, it has improved patients' health who had, there was a good success rate in treatment, okay, in Clostridium difficile infection. So we were mentioning a lot about probiotics and prebiotics too. So just to understand what are these different terms, is like probiotics are live bacteria which are used. It can be either as a capsule form or in a food form, but it should be live. So if the bacteria is dead, then it's not a probiotic. Okay, what are prebiotics then? Prebiotics are they are food materials which are given to the bacteria in the gut. So there are certain fiber-rich substances which can be prebiotics, which can promote the growth of the gut bacteria. So then what are the symbiotics? Symbiotic is just a combination of both the probiotics and prebiotics together and taking them. So just to reiterate, probiotics means live microbial food supplement. The bacteria should be live there. So whatever bacteria which you are using there should be non-pathogenic, non-toxic. Okay, so there are regulations in each of these countries for the use of the probiotics. There are a list of probiotics which are released by the government of or the Food and Drug Administration of each of these countries. So what microorganisms can be used as probiotics in products? So they should confer some sort of benefit when it's taken. And for prebiotics, as I told you, like they are the non-digestible food ingredients, means like when it is passing through the upper digestive tract, they should not be digested. They should not be hydrolyzed or absorbed, but it has to go into the colon where they will be utilized as food by the microflora there. Okay, and symbiotics, as I told you, is a combination of. You always see a lot of market products, okay, which have a probiotics claim, live cultures, most of them in the dairy products, where you say like this one has got a lot of um, bacteria, beneficial bacteria, especially from members of Lactobacillus, Bifidobacterium, and so on. But whether this is good or this is good, taking natural food on any day, taking a natural product is always better. Natural food is always better when compared to commercially available prebiotics. But what we need to also remember is like if your gut diversity is completely depleted, then to start with, it is okay to take with a probiotic. Means like the example is like if you want to throw apple seeds into a desert soil, is it going to grow? So you need to have some sort of fertility for them to flourish. So you can also start with some probiotics which are available in the market to improve your gut microbiota. But there is a conflicting report in this. Like some authors believe that, I mean, some researchers believe that uh, giving the probiotics actually it gives the opportunity of one or two or three types of microorganisms to multiply. Whereas if you depend on natural products, natural fermented products, something like sauerkraut, which is a sort of fermented product of cabbage and all that, which will have a lot of, it's a naturals, you have the commercially available sauerkraut and a natural homemade sauerkraut. So if you take homemade sauerkraut, you'll have at least 20 different types of microorganisms there. So that is more beneficial because it's diverse. Okay, so this is an exhaustive list of fermented products, which is from different countries, from Philippines, Vietnam, Thailand, and all. I'm not sure whether the font is big enough for you, for each of you to see. So I just want to overwhelm you with the idea that there are a lot of fermented products which are available all over Southeast Asia. This is a list from the products which are made from fermented fruits and vegetables from Asia. So which can be useful. It's not necessary that we need to depend only on the Indian type of food. Okay, we can look in for other options as well. Okay, and talking of the Indian food, like if you can remember the ragi-based fermented beverage, okay, which is served in Karnataka and Tamil Nadu, it's actually very rich in probiotic because it's mixed with curd, okay, and so and a lot of other products. You have products all over India, like from Manipur. This one is from Assam, and Dokla is from Gujarat. Okay, which are obtained from fermentation. You remember the famous song Uppu Karuvadu and Uravacha Soru? And what we are talking about, this Uravacha Soru is only the Palaya Sadam, actually the Niranjanam or the old leftover rice, okay, which is a practice which is done in Tamil Nadu and even in other villages in India. So you know that earlier days, no refrigerator. So what they do is like the leftover rice was left with it's being soaked in water and left in the kitchen counter. Next morning, what they do is like they add little curd to it or more buttermilk to that, and then they add little salt, onion, green chilies for taste. Okay, and also the rice kanji water actually, which is drained from the cooked rice. Okay, so they mix it and they allow it to ferment. Okay, that's a good source of probiotics. Okay, so by use of probiotics, they found this is one study which says like even in hospital care, use of the probiotics or the probiotic and prebiotic together as symbiotics has helped them to recover fast because they have found that the dysbiosis of microorganisms can even contribute to nosocomial infections. It can result in sepsis or organ failure. So, what are the ways to keep your bugs happy? Is you have to respect them, love them. Means you have to take care of them by eating healthy. There is also uh, a, a major belief that soil contributes a lot to the human gut microbiome. Okay, they have found that if you see the soil rhizosphere study and the human intestine, you see a lot of functional similarities between them. So what was what we do today is like our contact with the soil is largely reduced because most of us will live in apartments, and that's one major reason for the depletion of the gut microbiome. Okay, you know that there were habits of eating mud or some form of clay. It's also called as zeophagy in some in countries, some tribal population, especially in Africa, sometimes in India too. Okay, and researchers believe that this is very good for health because the clay, whatever they were consuming, had a lot of variety of minerals and also the diversity of microorganisms. So just to reflect and think, like in Hindu mythology, what we believe is like Krishna ate the mud, the clay, soil from this, and then we also have the biblical reference, or in the reference from Quran, that you have the manna falling from the heaven. Okay, and then actually manna was falling to the ground, and they picked it from the ground. So it means like along when they picked it from the ground, they picked also the microorganisms from the ground. So it's just a sort of reflection to think that our association with the soil to promote the gut microbiome is important. So don't get me wrong that I'm asking you to eat clay or soil for that matter, because I'm also a microbiologist, so I would not recommend to do that. But what I'm saying is like those days, like people were not that hygiene aware, or they were more free to eat natural food, in the sense like they go to a farm or they pick up fruit from the garden, they will eat it like that. Okay, which will also add on to the microorganisms, which will help in the diversity. But these days, like because of too much of sanitization, we have lost all these microorganisms. So what you need to teach your kids is like feed yourself with good microbes, eat bacteria. What I mean is like you take yogurt, you have lot of bacteria. You can prepare a lot of different probiotic foods which are available, which is rich in bacteria, which can be prepared. It not necessary it should be in your culture. It can be adopted from different countries. Okay, so you can eat bacteria. No need to over sanitize yourself. Okay, and we are already doing over sanitation because of the COVID-19, and you have to use antibiotics with justification, and most importantly, expose yourself with nature's microbes. The thing is, like, allow your kids to play with mud, soil in the beach. Okay, it's not only the kids; you also need to play. Okay, so if you want uh, to lead a healthy life, you need to have a lot of good microbes in your gut. So there are a lot of things which we already know. There are a few things which we do not know, which we want to know. And say, for example, whether microbes influence your food choices or appetite, or there is a question like if any new drugs or food chemicals are brought into market, whether they need to be tested first on gut microbiota, whether they are friendly with the gut microbiomes. So those are questions which are yet to be answered. Okay, so with that, we come to the end of this session. I thank you very much for the patient listening, and thanks once again to the organizing committee and the management of Dr. MGR Janaki.
Thank
You're
you,
most
sir.
welcome. Yeah. You're most
Yeah, thank
welcome
you.
to feedback me to my email, which is there, the slide there.
Yeah, thank you, sir. Uh, Thank you, sir, for such a sagacious presentation on the healthy interactions between microbes and intestinal epithelium. We have few questions from delegate participation. The first question is from Ms. Shobha. How can we protect ourselves from microbiome?
Protect yourself from microbiome. It means like you want to protect yourself from what you mean is like you don't want the bad bacteria. You don't classify them as bad bacteria and good bacteria. Most of these bacteria in your gut are doing good. But only thing is like you need to keep them in a diversified way. That's a very important. So to keep them diversified, what you need to do is like you have to eat diversified type of food, especially food, plant-based food, which are rich in vegetables, fibers, and so on. So that's what I told you in the earlier slides, that in the studies, what they advocated is like you need to take at least 20 to 30 different types of vegetables or fruits to be consumed, okay, within a week. Okay, so besides your, I'm not saying like you should be vegetarian. You can take your meat products, whatever you want, but in spite of that, you need to put in a lot of effort to eat a lot of plant products.
Okay, thank you, sir. There's another question from Mithuna. Are probiotic tablets effective?
Uh, Probiotics, yes. Like nowadays, when you go to the doctors, sometimes they also give you, along with antibiotics, they also give you probiotics. Actually, there is a contradictory view among researchers as such. One is like when you take antibiotics, anyway, your uh, gut microbiota is going to get reduced. So to replenish them, it is advised to take probiotics so that they supplement. That is one part of the theory. The second part of the theory, they say, is like if you are going to give your probiotics, the probiotics is what? Okay, it's a mixture of one or two organisms. It can be lactobacillus, it can be bifidobacterium, and so on. So it's just two or three types of organisms which are being enriched. It is not the diversity. So the other school of thought, what they believe is like instead of giving, if at all, if you want to start with, it's okay to take little bit of probiotics, but on a longer run, you need to diversify your microbial uh, community in your gut. So which means like you need to eat a lot of fiber-rich vegetable food.
Yeah, thank you, sir. Another question from Dr. S. Malati. How does gut microflora respond to food colorants?
That's one of the questions which I put it as a question mark in the conclusion. If you see, like, that's what is a major query. Like, it's not just the colorants, even the preservatives or whatever additives used to the food. Okay, they definitely have an impact on the gut microbiomes. So, anything which is not natural has to be avoided. Okay, but in a small quantity is okay. In a large quantity, it's going to affect the gut microbiome. So what researchers say is like in future, before you put any product or you find any food additive, you have to first test them on the gut microbiome. If you are happy that they does not alter the diversity of the gut microbiome, then you can go ahead with you adding them in the food. That's what is the recommendation.
Okay, sir. Another question, sir. Can you explain the mechanism behind the influence of microbes on obese and lean persons?
Uh, the mechanism as such, there are a lot of research which is currently going on. They are not able to exactly find why these things happen. But only thing what they trying to attribute is like uh, there are things like the diversity of the gut microorganisms. Actually, they some of them they actually lower your hunger. Okay, so the craving for food is reduced. So that's one reason that your obesity can be reduced because some people, and the other thing is like, as I told you, like how the gut microbes respond to the food. Okay, so I explained you with the example of a twin. For the same food, if it is rice, for one person there is a peak in the glucose level. For the other person, it's a flat curve. So why the microorganisms behave differently in different people is quite interesting and it's quite puzzling also. So a lot of research is going on. Like hope we will get some good answer someday.
Yeah, thank you, sir. Another question from Dr. N. Vijayalakshmi. What is the impact of microflora in vernal keratoconjunctivitis?
Microflora.
Yes, sir. The impact of
Okay.
microflora.
Can you repeat that again?
The impact of microflora in vernal keratoconjunctivitis.
Okay, so this is actually like what my lecture was mostly based on the gut microbiome. So we were talking on a lot of diseases, on protective natures the bacteria can offer to the, against the enteric pathogens and against the common diseases, NCDs, non-communicable diseases, and all that. But for infective uh, diseases in other parts of the body. Okay, we have a lot of other microorganisms in all the parts of the body, but not much is understood on that. But there can be a definite role on these organisms on the commensal flora against all the pathogenic infection. Means like any organ for that matter, if you have the commensal flora, okay, they are beneficial in preventing infection.
Okay, sir. Another question, sir, from Dr. Duga. What is the effect of microplastics that we consume through food? And does our microbiome have a defense to overcome that?
Microbiome will have a defense to overcome that, but we will not have a defense because if there are something which is inhibiting and hazardous for the microorganisms, a particular type of microorganisms will die, while the other group which can tolerate whatever chemicals or microplastics which are there will flourish. But that is not good for us. The microorganisms will survive anyway, but that is not good for us in the sense we always wanted the diversity in the microorganism. If one particular group alone proliferates, that is not good for us because that can result in some other clinical condition.
Okay, sir. Another question, sir. What is the impact of alcohol or cigarettes on these gut microbes?
Yeah, but yeah, alcohol because it's consumed through the uh, gut. Okay, so it'll have an effect. But in general, if you say smoking and drinking, all this will have a definite effect on the microbiome. So if you say like smoking for that matter, like it will definitely affect the microbiome in the respiratory tract, the commensal flora, which is actually protecting your the layers, okay, from the pathogenic invasion, from the pathogenic viruses, and so on. So there will be definitely lapse in the defense system if, for people who are smoking and.
Okay, sir. Okay, sir. Another question from Ms. Ashwini. Some fungal
When I
infection.
say drinking, yeah, when I say drinking, like it's not all. We are talking about alcoholics, but if some alcoholic beverages, something like wine, it's taken in minimum quantity, it will be beneficial because again, it's a fermented product. Wine is a fermented product. It's good for the gut microbes.
Okay, sir. Thank you, sir. Another question, sir. Some fungal infections cause eczema, which is influenced by dairy products. What about the role of gut microbiota in this case?
Uh, yes, as we studied, like uh, we were talking about the human milk oligosaccharides. If you remember, like I told you, like people who are not, the children who are not breastfed, they have a higher tendency to develop different conditions at a later stage. Similarly, like even like the babies which are born through C-section, it's all because of the diversity of the gut microbiome. So the effect of these gut microbiome to make you susceptible is there. Okay, to make you susceptible to certain allergic conditions, to make you susceptible to obesity, to make you susceptible to diabetes is there, but not necessary that you should get it. Okay, so we always say there is a 50% more chance say, for example, if you take a normal vaginal delivered baby and a C-section baby, there is a 50% more chance for the C-section baby to develop any of these conditions later in the life. Yeah.
Okay, sir. Okay. Another question, sir. What can a person do if he or she has lactose intolerance?
Yeah, there are a lot of other supplements for if you don't want lactose. That's what I told you. Like when we talk about probiotics, the most popular way is like using the dairy products. There are a lot of products based on yogurt and other things. But there are lots and lots of fermentation products which are available, which are natural. Plant-based or vegetable-based or fruit-based uh, um, probiotic. Actually that's there in the big list of two slides which I put. There are a lot of fermented fruits or probiotic fruits which are available. So they can be used. It's not necessary you need to depend on a dairy-based product.
Okay, sir. Another question, sir. What will happen to microbes if we fast for a day?
What will happen to the microbes?
If we fast for a day, the impact of fasting on gut microbiome.
Uh, Actually, it's just fasting for a day doesn't alter much uh, because you'll have enough stuff. That's what I'm saying. Like, but fasting for a longer time, basically, what is important is like even if you are not fasting, if you are taking food which is poor in fiber, uh, then that is not sufficient for the microorganisms to grow. Means like your gut microbiota is not happy with that sort of food. That's why they start eating your mucous membranes. So food, what the gut bacteria prefers is a fiber-rich food. So that's a thing when I say like you need to eat a lot of leafy vegetables, green vegetables. That's important.
Okay, sir. Another question from Ms. Akshaya, sir. Is MC4R gene, which contributes to obesity factor, anywhere related to gut microbiota?
They regulate in a particular way because there are a lot of studies which are done with which trying to link the association of these microorganisms, the products which are produced by them, and how they influence on the metabolism of the host. Okay, whether they influence expression of particular receptors, all those are being studied. So there are certain links which are established, but there is a long way to go because we still know that if there is a diverse microbial community, it's good. That much we know. Like, but why certain bacteria, in higher numbers, cause some harm in only certain individuals, but the same bacteria, when it present in other people, does not do it? Is $1 million question.
Okay, sir. Um, the next question is, what are the novel therapeutic approaches of type 2 diabetes based on the understanding of gut microflora host interactions?
Uh, as such, like if you see like in diabetes, like the not just the medication, they also say like your diet has to be regulated. That's a major advice is given for the diabetic people. So if you see like in general, the uh, the advice for the diabetic people is to take food which is rich in fiber. That's what is always recommended, rich in plant products, rich in fiber. So that is what the gut microbiome also wants. So in a way, it directly or indirectly, it's going to benefit the person.
Okay, sir. Another question, sir. Does selective manipulation of the gut microbiota improves immune status?
Yes, that is the thing which people are working now. Like when you go to take your babies in future to for a check, it's out of the regular check of height, weight, and all. Like there will be also check on the gut microbiome. They will take fecal material and do screening, okay, of the gut microbiome and see what microbes are there or what are not there and how to supplement them. So that will be the future. Yes.
Okay, sir. Another question, sir. What is the association between Helicobacter pylori infection and gastrointestinal malignancy?
Some helicobacter infections can become cancerous, and we have known that certain conditions like can convert into a malignancy. So, but whether it is because of the organism, because as I told you, Helicobacter is a commensal flora, so it's there in the gut of everybody. But why it causes ulcers in few people and the other forms of cancers in other people, it's again debatable, and that is because of the dysbiosis. That's what they say. Like when the symbiosis of the gut microbiome and the host is upset for some reason, then because of that it can cause infection. Similar to like how dandruff uh, occurs, like means like uh, it's caused by Malassezia species. Like it's there, it's a commensal flora which is present in the scalp of everybody. But why it causes dandruff only in certain people? The $1 million question.
Okay, sir. Another question, sir. Does microbiome diet introduced by Raphael Kelman improve the gut microbiota and reduce obesity?
Uh, what they mean is artificial microbiome introduction or by natural means.
Okay. One more question, sir. Can gut microbes be promising targets for the management of type 2 diabetes?
Yes, of course. Like now, when we talk about too much of these microorganisms, like how we have been hearing about the COVID virus from time to time for the last few months, like we always say it's a pandemic of virus and a epidemic of information about the virus, something like that. After this human microbiome project was done, there are a lot of studies happening all over the world trying to uh, associate every possible disease with the gut microbiome. So there are other researchers who have a different school of thought. Is say like most of these studies they are trying to relate sort of a Me Too studies, so that they want to relate everything to the gut microbiome. So that is also there. That sort of criticism is also there.
Okay, sir. Another question from Mahalakshmi Sridharan, sir. Does gut microbiota have effect on congenital diseases?
Uh, yes, it can be, because some of these diseases, because they say like some diseases are diseases of the mental health and all can be congenital, and they have found that association of the gut microbiota to some of the mental health conditions. So yes, possibility is there, like, but a lot of research has to be done.
Okay, sir. One more question, sir. What is the role of pattern recognition receptors in mediating the bidirectional crosstalk between microbial symbionts and their host?
Lot of research is being done on this. Like uh, on a broader perspective, what you need to understand is like because if you look into what microbes give, what benefit, and how it works, whether you can manipulate by using a microbe, you can manipulate the human system by altering this microbe, and all that is again debatable. Because if you ask me, as a person, my individual opinion is like I would always prefer a sort of a holistic approach. Means like you need to find a way to diversify your microorganisms in the gut. That's important. Okay, so that's a way for life rather than trying to introduce specific microbes for specific things. That's still debatable. It's a long way of research to go.
Thank you, sir. And one more question from Ms. Yogalakshmi. Does coffee consumption affect the gut microbiota?
Coffee, no, in certain limited concentration, I don't think it's going to affect because, well, as I told you, like gut microorganisms also prefer a variety of food. So I don't think coffee is a problem there.
Okay, sir. And one more question, sir. Can islet of Langerhans be promoted by the intake of prebiotics or probiotics?
What can be promoted?
Islet of Langerhans cells.
Okay. See, as I told you, like this all what is happening is a sort of holistic approach. So when we say like we cannot totally ignore the modern medicine, the drugs which are there in the modern medicine, which is used for different conditions like diabetes, obesity, and all that. So I don't think like we are just going away with it and then starting to go towards the microorganisms. It should have sort of a complementary approach where both should go hand in hand, where the modern medicine will also be there, and wherever it's necessary, you have to supplement with probiotics and prebiotics.
We are moving to one last question, sir. Does pills taken for depression and stress have a negative impact on gut flora?
Actually, the question of the gut and the brain response. Okay, so it's actually still not very clearly understood whether because of the dysbiosis there is a stress or anxiety, or there is dysbiosis when there is a stress. What I mean, whether it's a vice versa process. So we see like whenever there is a stress condition or anxiety, you see a dysbiosis of the gut microbiota. So, but what people believe like is like by sometimes by improving the gut microbiome, it helps to bring back the normal mood. So that way it's linked. Yeah.
Thank you, sir, for making us realize that microbes and gut epithelium are more than bickering neighbors. Your years of research, depths of understanding, and presenting your subject matter expertise in an intrigued manner produce one of the most knowledgeable sessions in our webinar series today, sir. Your presence has definitely enriched our webinar. A heartfelt thanks once again, sir.
Thank you very much, and goodbye to all of you.
On that note, I would like to give a gentle reminder about tomorrow's session. Is my immune system a friend or foe? To shed light on this, an erudite speaker, Dr. Gauri Shankar Rajan, Associate Principal Scientist, Global PPDM, Merck & Co., USA, will connect with us at 5 p.m. tomorrow. Kindly join us. Thank you all.